Gene editing is a revolutionary technology that enables scientists to modify the DNA of living organisms, including plants, animals, and humans. It involves making precise changes to an organism's genetic material, allowing researchers to add, remove, or modify specific DNA sequences. Gene editing has profound implications for various fields, including medicine, agriculture, and biotechnology. One of the most powerful and widely used gene editing techniques is the CRISPR-Cas9 system. 1. CRISPR-Cas9 system. The CRISPR-Cas9, clustered regularly interspaced short palindromic repeats, CRISPR-associated protein 9, system is a molecular tool that was adapted from a natural defense mechanism found in bacteria. It allows scientists to target and modify specific DNA sequences with unprecedented precision and ease. The CRISPR-Cas9 system consists of two main components. Guide RNA, gRNA. The guide RNA is a synthetic molecule that is designed to be complementary to the target DNA sequence. It guides the Cas9 enzyme to the precise location on the genome that needs to be edited. Cas9 enzyme. Cas9 is an enzyme that acts as molecular scissors. It cuts the DNA at the target site specified by the guide RNA. Once the DNA is cut, the cell's natural repair mechanisms come into play to either introduce specific changes or create knockout mutations by disrupting the targeted gene. Gene editing process. The gene editing process involves several key steps. Designing the gRNA, scientists design a custom guide RNA that is complementary to the target DNA sequence. This is a critical step, as the accuracy of the guide RNA determines the specificity of the gene editing process. Delivery of CRISPR-Cas9, the guide RNA and Cas9 enzyme are introduced into the target cells. This can be achieved using various methods, such as viral vectors, nanoparticles, or electroporation. Target DNA cleavage, the guide RNA guides the Cas9 enzyme to the target DNA sequence. Cas9 then makes a double-stranded cut at the specified location. DNA repair mechanisms. After the DNA is cut, the cell's repair mechanisms come into play. There are two main repair pathways, non-homologous end joining, NHEJ, and homology-directed repair, HDR. NHEJ, this repair mechanism is error-prone and often introduces small insertions or deletions at the site of the DNA cut. These changes can result in gene knockout or disruption. HDR, this repair mechanism uses an exogenously provided DNA template as a guide to repair the cut DNA. It can be utilized to insert specific sequences or correct mutations with high precision. However, HDR is less efficient than NHEJ. Applications. Gene editing has a wide range of applications. Medical research and therapy. Gene editing can be used to study the function of specific genes and their role in diseases. It also holds promise for treating genetic disorders by correcting or replacing defective genes. Agriculture. Gene editing can improve crop yields, enhance nutritional content, and increase disease resistance in plants. Livestock improvement. Gene editing can be used to produce animals with desirable traits, such as disease resistance, improved meat quality, and increased milk production. Biotechnology. Gene editing can create organisms that produce valuable compounds, such as biofuels or pharmaceuticals. Ethical considerations. The power of gene editing also raises important ethical concerns, such as off-target effects. CRISPR-Cas9 may introduce unintended changes at other sites in the genome, potentially leading to unintended consequences. Germline editing. Editing genes in human embryos, sperm, or eggs. Germline editing raises concerns about passing modified genes to future generations and the potential for unintended long-term effects. Designer babies. The ability to edit genes could be misused for creating designer babies with enhanced traits, leading to ethical and social implications. Biodiversity. Gene editing in agriculture could impact natural ecosystems and biodiversity. Gene editing, particularly the CRISPR-Cas9 system, has revolutionized biology and offers immense potential for scientific advancement and practical applications. However, the technology also demands careful consideration of ethical, safety, and regulatory concerns as its use becomes more widespread. As research and technology continue to advance, the full impact of gene editing on society and the environment will become more apparent.